The aim of the following experiment is to measure the contractile activity of cultured muscle fibers in response to stimulation in a controlled in vitro environment. This is achieved by plating dissociated muscle cells on microscale silicon cantilevers and inducing the fusion of these mononucleated cells to form fibers termed myotubes. These cantilever chips, supporting differentiated muscle cells, are then transferred to an electrophysiology microscope modified to support a laser and photodetector used to measure cantilever deflection in response to muscle contraction. After placement of the chip in the rig, the laser is positioned so that it focuses on the tip of the cantilever, and the photodetector is likewise aligned so that it catches the deflected beam. Results are obtained that show the degree of cantilever deflection, which is caused by muscle contraction, in response to electrical or chemical treatment, providing a direct measurement of the contraction profile of the seeded cells in real time. Ultimately, cantilever displacement can be processed to provide a readout of myofiber force. This system is multiplexed to provide a potential high-throughput testbed for assessment of changes in contractile function in response to drug treatment, disease state, or exercise protocols.
The main advantage of this technique, compared with existing methods such as patch clamp electrophysiology, is that this method for functional analysis is non-invasive and easily adaptable for high-throughput studies. This method can help answer key questions in the drug development field, such as identification of appropriate dose-response concentrations prior to clinical evaluation.
The implications of this technique extend toward therapy of multiple muscular diseases because it provides means to investigate the functional performance of cells from different pathological phenotypes.